Well, good afternoon, everyone, and on behalf of the College of Engineering, I want to welcome all of you. Thank you for joining today's College of Engineering Distinguished Lecture Series. And I'm absolutely delighted to present this afternoon's speaker, Dr. Deirdre Meldrum, Senior Scientist and Director of the Arizona State University Biodesign Institute Center for Biosignatures, Discovery, and Automation. So I want to spend a few minutes telling you about her very distinguished career. Her talk today, Biosignatures, Automation for Improved Human Health, will address a desire that we all have, which is to have great chronic good health. The analysis and adaptation of personal biosignatures would enable a healthcare system that focuses on people healthy, keeping them healthy, and reducing the time that sick people spend in hospitals and in clinical care. Disease risk could be prevented and controlled, and thanks to interventional therapeutic treatment of individuals based on their biosignatures, the overall healthcare picture would change. Now, Dr. Meldrum is a visionary leader of science and technological advancement, and she has spent her career blurring the traditional boundaries of engineering and science and creating new paths for discovery. She's known for her ability to integrate multiple disciplines in engineering, from civil, electrical, chemical, to material science, with fields like biology, nanotechnology, genomics, oceanography, and environmental studies. Her research interests include gene automation, microscale systems for biological applications, ecogenomics, robotics, and control systems. She has helped design systems and equipment to explore outer space, the bottom of the ocean, and the inner working of cells. She'll go to any length or depth in pursuit of her research. In August of 2006, as part of the team on the ocean research vessel Atlantis, she descended in the Alvin submersible to the floor about 2,200 meters below sea level to perform experiments at Endeavour Ridge in the Northeast Pacific Ocean. I don't know about you, but I'm ready to go work for her. A recent issue of Phoenix Woman magazine, in an article on Arizona's leading scientists, referred to Dr. Meldrum as, quote, the new breed, end of quote, in great part because of her impressive work ethic. At the time, she was both director of Arizona State University's Center for Ecogenomics and the Biodesign Institute. That wasn't enough. She was also the dean at ASU School of Engineering, a position that she held from 2007 through 2010. Cannot imagine how she did all those jobs. Dr. Meldrum defines ecogenomics as working in a harsh environment, the human body at one extreme, oceans and space at the other. Her research ranges from analyzing how living cells work to growing bacteria intended to be used in the creation of biofuel. Genomics, as she points out, is a cutting-edge green discipline. Fascinating definition. As a high school student, Deirdre Meldrum wanted to be an architect. Fortunately for us, a summer stint with the Junior Engineering Technical Society encouraged her natural talents in math, science, and problem solving, and ended up at the University of Washington, where she majored in civil engineering. More significantly, she did some co-op work at the Puget Sound Naval Shipyard in Bremerton, Washington in '79, and then in NASA at the Johnson Space Center in '80 and '81, where she helped train astronauts on the shuttle mission simulator. In '85, she earned a master's degree at RPI, studying robotics and control systems. That led to a stint at Pasadena's Jet Propulsion Lab, where she worked on the Galileo spacecraft. After earning a PhD in electrical engineering at Stanford in '93, she joined the faculty at the University of Washington, where she embraced the Human Genome Project. Because of the project's size and scope, demanded automation, which encouraged Dr. Meldrum to apply robotics to the investigation. Her subsequent work in an entirely automated research instrument dubbed Acapella attracted the interest of, and more importantly, the funding of, the NIH, which honored her with a Special Emphasis Research Career Award. She founded the University of Washington's Genomation Laboratory, and in 1996 received the Presidential Early Career Award for Scientists and Engineers, recognizing her innovative research utilizing a broad set of interdisciplinary approaches to advance DNA sequencing. In 2001, she was awarded an $18 million grant for a National Institute of Health Center of Excellence in Genomic Science, which led to the establishment of the Microscale Life Sciences Center. The grant was renewed for an additional $18 million in another 5 years in 2006. As a member of the University of Washington's Regional Scale Nodes Project, she became involved in the goal of constructing a cabled underwater observatory to be built on the Juan de Fuca tectonic plate in the Northeast Pacific Ocean, for real-time observations and experiments with sensors. She is a member of the National Advisory Council for the Human Genome Research, the U.S. Department of Health and Human Services. She's a fellow of the IEEE, a fellow of the American Association of the Advancement of Science, has 6 patents and more than 150 articles and books and peer-reviewed journal papers. She's a popular speaker with recent appearances at the 10th IEEE conference in robotics and automation and at the 2010 National Academy of Engineering Grand Challenges Summit, which in fact she chaired. Her opening remarks at the later event stressed the need for scientists to focus on the progress necessary to maintain and improve the quality of life in an increasingly complex world. Last-minute saves from catastrophe, she warned, exist solely in science fiction stories that find heroes scrambling to avert rampaging viruses or global-scale natural disasters. It doesn't happen that way in real life, she said. The most effective ways to ensure health, safety, freedom, and prosperity for the future are a lot less dramatic. It takes years of committed, coordinated, and sustained effort. End of quote. One of Dr. Meldrum's top priorities is to prepare the next generation of engineers so they can excel in a global community. She has lived the mantra she passes on to young people looking to break into our field. Be as involved as possible, get to know your teachers, enter science fairs, and take advantage of summer programs and opportunity. She well remembers her inquisitive and enthusiastic younger self and has encouraged local high school students to work in her labs at Arizona State. It's a great pleasure to welcome Deirdre Meldrum to UC Davis. Join me in giving her a warm welcome. Great to have
you. Wow, I've never had an introduction like that before. I don't know where you were able to learn all of this. Anyway, thanks everyone. It's a real pleasure to be here. I've been really impressed with everything I've seen today, the people I've met with, so a really great working environment and a real pleasure to be here. As you know, I do work on several different things, and I'm going to keep this talk focused entirely on human health and disease. But if you're interested in something towards the end, if we have time, I'd be happy to discuss any other things that you're interested in. Can everyone hear me? Actually, I think I didn't turn this one on. Does that help? Good, because I can be hard to hear sometimes. Okay. So what I'm going to talk about is, in the larger scheme of things, is a Biosignatures initiative. And so let's first make sure everyone understands what we mean by biosignature. And it's all the biological information about you that would be predictive of your future health status. So it could be your genomic information, proteomics, cellomic imaging, behavioral information, and so on. And figuring out what of that will give you a prediction of your future health. And this is a big effort that we think could scale to something on the order of a national laboratory. That's a big undertaking, but it could conceivably be something like a Lawrence Berkeley National Lab. And the whole goal is to really change our whole focus of how we think about health. Instead of treatment of sick and dying, to how can we figure out and do pre-symptomatic diagnosis and maybe even interventional treatment before you have any symptoms. You can imagine that's a very hard thing to do, especially culturally. We're not used to doing that. You don't do anything unless there's something evidently wrong. But if we can figure out ways to do this pre-symptomatic diagnosis, you could change the course of your health so that you can live longer and healthier lives. So there's a lot of aspects to this that are not only the technology, but of course policy, insurance, you name it. It's a very huge undertaking. So we're thinking about this in terms of national scale laboratory. I did go around and visit about 8 different national laboratories. Those are mostly the ones you think of, Department of Defense kinds of laboratories, or Department of Energy. This would be something totally different and focused on health. And would need to be something that partners with, say, the FDA, NIH, Center for Disease Control, and other entities. And also have the ability to build up not only disease-specific biorepositories, but normal biorepositories. And what does that mean? And then there's a whole pipeline to this, which I show here. So there's the discovery of what the biosignature is. And once you've discovered what that biosignature is, and there can be multiple ones, then standardization of the biosignature, qualification, and clinical validation. And so all of this really requires a large amount of automation. So once you have the discovery phase, you really have to put this into something so you have reproducibility, reliability. You can perform all these tests using the samples from the biorepositories. And one reason for thinking of this in this, like a national scale laboratory is because you need to have that consistency of all these different experiments. It's very true if you talk to someone that does an experiment in one laboratory, they can't necessarily repeat it at another one. And so there's a lot of things there that need to be standardized and qualified. So this looks a little bit complex, but I'll walk you through it. This is how we're thinking about the biosignature. And I'm focusing here on the discovery. I'm not going to go into those other parts of it for this presentation. So let's say you have your current health status. That could be information in your medical record, maybe your DNA sequence and other information. Then you could figure out some disease risk vectors, go into some preventive strategies, early diagnosis, and some kind of therapies. Well, the different aspects of the biosignature, as I mentioned, the genomics, proteomics, cellomics, imaging, and outcomes, and I give some examples here of what kinds of information that could be. You probably hear a lot about people saying a biomarker for this or a biomarker for that. We think of a biomarker as being just one component of the more comprehensive biosignature. And those on the left-hand side, these are mainly screening kinds of tests that could be done on these normal and disease biorepositories, the non-invasive typically, whereas these ones on the right are more interventional treatments and outcomes. Now, if we look at some diseases, let's look at cancer, just to give you an example. Ideally, this biosignature would be predictive of all kinds of different diseases, not just one. But we have to start in a simple manner and figure out, well, what is a biosignature for some kind of disease? So let's look at cancer. And you can see here lung, colon, breast, and prostate. Well, what's striking here is that the number of deaths from lung cancer, that's the red bar, is more than the other three combined. Do you have any idea why that is? Smoking. The key thing is that there are screening things that you can do for colon, breast, and prostate. There are no screening programs or anything for lung. And typically once lung cancer is detected, it's too late. And most people die from it. So if you look at this, lung cancer is the #1 cancer killer in the world. 2 million lung cancer deaths worldwide, large number in the US. Then we're looking at Arizona and launching a screening trial in the state of Arizona. There's 12 lung cancer deaths per day in Arizona. That's not really taking into account the Native American population because we don't have very good data on that. So we believe that there's need for screening programs, and there was the National Lung Cancer Screening Trial that finally came out. Actually, Harold Varmus stopped it early because it was already showing that with CT screening that there was a 20% improvement in reduction of deaths from lung cancer by doing CT screening. And then just one more thing to make sure everyone's on the same page, to make sure you understand a false negative rate and a false positive rate. So the false negative rate is the number of people in a particular disease that would not be identified, whereas false positive is you're erroneously identified as having a disease and then may go on to have some treatment that you don't need. So both of those we don't want those to have false negative rates that are very high or false positive rates that are very high. And the reason that's important is we're trying to come up with some way to predict your risk of having a particular disease. Now let's just say, let's go with the lung cancer. We're selecting lung cancer as our demonstration project for the Biosignature initiative and have experts in lung cancer that also do CT, and we're developing other technologies to come up with the Biosignature. So in lung cancer, some examples of ways that you might come up with the biosignature, and we haven't done this yet. We're in the midst of trying to define that. But one example would be some kind of mutation, DNA mutation, and it may have an 80% false positive and a 50% false negative rate. The false positive rate is so high because normal people have this mutation. So if that was your only biomarker, you wouldn't be doing a very good job of prediction. Likewise, if you look at angiogenesis pathways and so on, there's a high false positive rate because of inflammation, so that affects the result. Cellomics, we actually have a good handle on some approaches, which I'll share with you. When you go to CT imaging, well, if the tumor is smaller than 4 millimeters, you're going to have a 100% false negative rate because you can't even detect it. But once it gets big enough to detect it, you still have a high false positive rate because 50% of the tumors are non-cancerous. So if you're just going with one bit of information, you can see here that you may not have a very good prediction. So keep that in mind, because then as we go here, one thing you can do is in the biosignature, take all this information and integrate it in some smart way. I mean, one simple way is multiplying. We don't know if that's really the right way to do it yet. But the idea is that you combine all this different kinds of information in some way that gives you your prediction and has low false negative and false positive rates. So what I'm going to do is walk you through some of the different technologies here for biosignature discovery. Genomics, we're not working in that area per se, but I give some examples from people at ASU, Stuart Lindsay, and where is the one that worked with Stuart? Oh, no Stuart. Anyway, he's been doing some next-gen sequencing with electron tunneling. That's been a really successful program in the NIH to rapidly improve genome sequencing technologies. The rate at which sequences can be obtained and the cost has just been dramatic. It's much better than a Moore's Law kind of an approach. And I'll just also mention here that George Church at Harvard has a personal genomes project, and I mentioned that to some people today, is where people can volunteer to have their genome sequenced, and then everything's put out in a public database and combined with medical records and other things for people to study. So you'll see more and more of that as well. Then in terms of proteomics, there are a lot of different approaches and new technologies. Josh LeBaire at ASU does a lot of work. He's developed a way to get high level of protein expression using his NAPA technology. It's a nucleic acid programmable protein array. He's done a lot of studies and come up with specific biomarkers, mostly for breast cancer and diabetes. Certainly it can be applied to other diseases as well. Now I'm going to spend a lot of my time talking about cellomics and two different approaches. One is for looking at cell morphology, and then the other one is looking at cell physiology. And we think we can gain a lot of information there that can be part of the biosignature. The morphology is utilizing a technology called the cell CT, so computed tomography. It's developed by a company called VisionGate. We have the first instrument of that in my laboratory, and this company was moved from Seattle to Phoenix by the mayor, by investment from the mayor in the city of Phoenix. And this is really a novel technology that I'll describe, and that is taken in my lab. So first of all, what we're looking at is alterations in a cell at the nuclear and cellular level to see how a cell changes through its course of, say, neoplastic progression. So if you look at the normal cell on the left and then a cancer cell on the right, you can see all kinds of changes in the nuclear membrane, the nucleolar size, chromatin pattern, nuclear lamina, and so on. And the cell CT can currently quantitate over 500 different features, which is pretty amazing. In fact, if you go to what is typically done, so sputum has been shown in the past to not be very effective or useful for detecting lung cancer. And that's because they just take sputum and they smear it on a slide. And do you think you could look at that and diagnose lung cancer? It'd be pretty difficult. But honestly, all cancers except one kind are done in a way that's done by a pathologist, making the call without any quantitative measures. They just look at it. The only test that's done in a quantitative way and it's in an automated instrument is the pap smear. And that's with an instrument that was developed by a company called Neopath that was then acquired by Becton Dickinson, and it's being used worldwide for diagnosing cervical cancer. So just for example, in the sputum, there are a lot of reasons why it doesn't work well, but collection is one thing. So there's a lot of different factors here. How is the sample collected? how is it prepared, and then how is it analyzed. And so you can see here there can be overlapping cells. They can be, in fact, I'll give you some examples of what can happen in the presentation of the cell. But then, of course, if it's the human pathologist making the diagnosis, there's issues there as well. They're highly trained and they're very good, but we can do better. In 2D cell analysis, so most the analyses are still done in a two-dimensional fashion. And if you were to look at these cells, you would call the one on the left normal and you would call the one on the right abnormal. And it actually is the same cell. It just depends on how it was presented and where you're looking in the focal plane of that cell. So that's one factor. You can see here it just depends on where you're looking. The other one is the orientation. So in all of those 3 cases, you would call it normal, but in the upper right, you can actually see there's a chromosome deletion and a mutation, and you might very easily miss that. And I should use this really cool pointer, thank you. So the idea is to go instead from 2D to go to 3 dimensions. And this is an optical projection tomography. It's inspired by radiological X-ray CT, but instead uses photons instead of X-rays. And the idea is to compute the true 3D image with isotropic resolution and the ability to compute all kinds of different quantitative features. And it's done by putting a cell in a capillary tube and then producing an analysis so that you can actually automate this whole thing. So it's CT where when a human goes in for CT, the instrument moves around the human. Fortunately, you're not getting rotated around like this. Well, here a cell is small, so we rotate the cell inside the capillary. And so there's a lot of technical features that were developed here with the lens and being able to quickly go through the different focal planes, its visible light, and then rotating the cells. And I'll just show you how this works. You put cells into a capillary and you rotate the capillary, and then you do these pseudo projections through the focal plane here. And when the cell rotates, you do 500 images around the cell. And this instrument, the one in our lab, I must say, is on the slow side. Their newer version will be able to analyze a whole sample, which means the processing of it and analysis of 700 cells and the diagnosis in 15 minutes. So it's been improved dramatically. And then you can see here what happens is you do these different projections and then you do the reconstruction to get the 3D image of the cell. You can do it in absorption mode, and they also have fluorescence mode. And I'm going to show you some pretty images, and those are really for the human to see and visualize, but they're unnecessary because everything is quantitative and there's all the data of that. And then the coloring that I'll show you actually is just coloring that's done after the fact to correspond to increasing density of the DNA. So there's what you get there, and you can see that chromosome deletion right there. That's a future vision of what it's going to look like. So here, the 2D can't see a whole lot, but now when you go to the 3D, and that's just a slice through it, you can see a lot more features, and I'll show you some examples. But before I do that, just to discuss something about what I was saying about the pathologist and then this automated instrument. The company put a group of pathologists in a room, highly experienced pathologists, and had them do all kinds of readings of samples. And the best the pathologist can do is 72% sensitivity, whereas the automated instrumentation in 3D is already up here, and as you incorporate more and more quantitative features, it approaches 100%. So this is some examples, and I'll go through some ways, but we think this is a highly promising way to get some of the biosignature features in terms of cellular morphology. These are fixed cells. These are not live cells. Here's an epithelial cell from the lung, And that's looking from the outside. That's the nucleus. And then we can look inside, and they like to say those are Bart from The Simpsons hairdo. So you can see inside here in the normal cell. So those are different chromosomes, the chromatin structure, all kinds of different features there. This is a cell that is adenocarcinoma. And one classic feature is it has a very enlarged nucleus. And they can measure this enlarged nucleus, and you just saw that little blip. That's the chromosome deletion. And then if you look inside, it's amazing. Seeing things that people thought were there but hadn't seen in this fashion. One is the nuclear membrane and all these invaginations. You see all kinds of structural features that you can quantify. And here's another one, squamous cancer cell. And what's important, especially if you're going to have use something like this for diagnostics, is to figure out your specificity and your sensitivity. That's the same thing as your false negative and your false positive rates, or 1 minus. And so then you want to get that up as high as possible and figure out where you're going to operate in terms of sensitivity and specificity. So you could be anywhere up in this range. And this is really excellent. This is the curve for one single cell in the instrument. So there have been clinical trials done and studies to demonstrate that to make the call for a diagnosis for one sample requires analysis of 700 cells. And that's actually sputum produced over 3 consecutive mornings. The first morning produces some of the cells. The second morning is the best. It's gotten everything going. And then the third is just for good measure, in a sense. And those cells that are produced in the sputum can even get the cells from way deep inside the lungs. So they think this is a very good test. And then you can see, as just like the pap smear test, you would typically do it every year, and that would make it so that you don't miss someone after, say, 2 to 3 years. It depends on what your sensitivity is. So here, let's say you did these tests annually, you would be able to catch this person's lung cancer. And if this were implemented in a screening program, you could save a lot of lives every year, because the sooner you can catch the disease, there's more likelihood you have of actually curing the patient. In fact, there have already been studies shown that you can actually detect metaplasia and dysplasia, which is precancerous. And if you have that, you can treat the individual with anti-inflammatory drugs, and they don't go on to get cancer. So that's one avenue for treatment. And just to give you some examples of some other kinds of cancer. Pancreatic. And you'll see here through these examples that they have different features as they go from being normal to cancer cells. This is an interesting case in which this is a macrophage that's taken from sputum, so it came out of the lungs. And you wouldn't normally find lipids in there, but what's happened is this individual probably had some acid reflux or something, and the macrophages ate it, and it was found in the lungs. So you could even detect someone that has GERD, gastroesophageal reflux disorder. So, so there's many applications of all of this. Now I'm going to go to an example, which is an area that we've actually been doing a lot of studies with this instrument in our lab, and also with our physiological studies, and that's to look at Barrett's esophagus and esophageal cancer. And this is with Brian Reed at the Fred Hutch Cancer Research Center. And also more recently, we've been getting primary cells from physicians at the Mayo Clinic. But we have all these different cell lines, the normal, metaplasia, dysplasia, and also adenocarcinoma. And what happens in an individual with Barrett's is that they start getting what's called Barrett's esophagus, which you can see here. And then they start forming these crypts in the structure of their esophagus. And Brian Reed and his team have already shown there's great heterogeneity in the cells in these crypts, and we've been doing studies on them for the cell physiological measurements. They've also, what Brian does is he's tracked patients for decades and taken, and in a very sequential fashion, biopsies. They're rice grain sized biopsies up and down and around the esophagus. And then they do extensive DNA and other kinds of analyses on them. So they have this wealth of information that we can also now correlate with some of the things that we're doing in these cell-based studies. In particular, correlate changes in the morphology with the genetic alterations. And so in the cell CT imaging and the stuff that we've been doing in our lab, we developed some of our own features and image processing approaches on the cell CT instrument. And I'll just show you here some of our images. So you can see here how the nucleus changes over these different kinds of cells, metaplasia, dysplasia, and carcinoma. And so you can see here that that could be a very distinctive biosignature for these different stages of neoplastic progression. And we also can correlate these with the genetic alterations. So that gets you into the genomic part of the biosignature. And this just gives you an example of some of the features: nucleus to cytoplasm volume, the number of the nucleoli, total nucleolar volume. We can even measure the chromatin texture, smoothness, and so on. And VisionGate has over 500. We're only using like 40 some in this work. We've also, we're part of a team, it's called the Physical Sciences Oncology Center. Have you heard of those with the National Cancer Institute? Where they are trying to bring scientists from other disciplines, like the physical sciences, to address and see what they can come up with in terms of understanding cancer. And so we're on a team to do that and looking at the morphology. And some of our studies here are with some of the scientists at UCSF. And so these are breast cancer cells from them. And when we put them in the cell CT, they look very different than the Barrett's epithelial cells. In fact, the nucleus gets a very kind of squashed appearance and other features which are very unusual. And what we are seeing is that all these different cell types have different things that happen to them in this progression towards cancer. Okay, now I'm going to move on to the other part of cellomics that we're working on, which is the cell physiology. And this is taking live cells, single cells, and doing stimulus responses experiments on them. So the stimuli could be something biochemical, an infection, or some kind of temporal cue, and then looking at the outcome. So we do this in a controlled microenvironment. And the idea is to understand the dynamics of the cell and really understand the different states: before stimulus, what's happening over time, and then some post-stimulus. So you can imagine in the case of Barrett's, just even having a change in the pH in the environment, how does that change the outcome of the cell? So what we're interested in doing is using this technology to measure different parameters of the live cell, to understand different pathways of cell life and cell death, and figure out why some live and some die. So there's non-inflammatory pathways, but also pro-inflammatory. This is pyrotosis, and we work with Brad Cookson at the University of Washington. He's in the medical school there, and also in laboratory medicine, on understanding this pathway. And then I already mentioned Brian Reed and the neoplastic progression pathway, or cell proliferation. So if you have a single cell on a device, and I'll show you a little bit more about that, there are different measurements that we like to be able to make on the live cells: oxygen, pH, membrane potential, all kinds of different things. And then fix the cell and take measurements of the state of that cell, the DNA, RNA, and proteins. So we've been developing technologies for that. The single-cell proteomics work is by Norm DeVicki, who was at University of Washington. He's now at Notre Dame and does a two-dimensional capillary electrophoresis for single-cell proteomics. We've been focusing mostly, the late PCR is work with Larry Wong at Brandeis University. That's a linear after the exponential, and you can look at ratios of different parameters and get loss of heterozygosity and other genomic features. And then transcriptomics is another area that we've been working on, and I'll mention a little bit about that. So let's go back to the Barrett's example. And we've done a variety of tests, both in batch chips, where we have thousands of cells, and then look at the single cells. And there's tremendous heterogeneity in all the different experiments that we run with these cells. And we're looking at things like the oxygen consumption rate, mitochondrial mass and potential, and then also looking at all these different cell lines. And as I said, Brian has all this information about the genetic components and even outcomes of these individuals. And so we're trying to make different correlations there with the information. This just is to show you what the oxygen consumption rate measurements look like for the CPA cells, the metaplastic, for these thousands of cells. And these rates are lower than what you would see in the single live cells. Oh, and these are done with our oxygen sensors. It's a platinum porphyrin. We've come up with a variety of different ones, but most of our sensors are immobilized in a polymer, and we have different reporter chemistries. So they're all fluorescence or optical sensors. This shows you what single cell oxygen consumption rate measurements look like. OCR is oxygen consumption rate. And these are the CPA and the CPC cells, the metaplastic and dysplastic. And you can see how it varies dramatically for all these different single cells. Another thing we were looking at is the mitochondrial mass. And here, these are the normal cells. There's less change there than you have in these other cells that are progressing towards cancer. And then you can also see how it correlates with the genetic change. I mentioned we were also working with Brad Cookson on pyrotosis, and this is just a diagram showing this. And a lot of the temporal order of these different events in pyrotosis are unknown. So it's caspase 1, there's pore formation, ion flux, release of inflammatory cytokines, and eventually permeabilization of the cell membrane and release of the cellular contents. So trying to understand this process. And so you can imagine being able to measure some of the things that we've been developing will help us get at some of these pathways. And actually, we're working with Brad now on a systems biology effort to really get at more of these processes here with our technology, but also incorporating modeling. Another thing we're very interested in is in cellular metabolism, and oxidative phosphorylation is generally what's used to generate the ATP in a normal cell, but in a cancer cell, it changes to a glycolysis process, and there's increased uptake in the glucose, decreased oxygen, and then extracellular acidification here. And so we're interested in measuring these things like glucose, ATP, oxygen, and pH, so that we can understand these processes and what happens as the cell transforms. So, I guess I didn't need that. I'm going to focus right now on this part here in our technology. We've developed the capability to make arrays of these chambers. So I just show one here, and to put sensors in there, and then to have a hermetic seal so that we can do these drawdown experiments and measure oxygen consumption rates and other things. And these show the platforms. They look kind of big, but that's because they're on laser scanning confocal microscopes in microenvironmental control, and then they have the optical sensing capability, and that shows you the example of one of the chips with different, that one's oxygen sensors. And then, of course, we want to be able to automate and get higher throughput of these, and towards the end I'll show you the next generation system that looks much more sleek than these massive systems in our labs. But the idea here, we can put multiple sensors in the device. We can do things that are spatially separated and also spectrally separated, and then certainly have arrays of these on a chip. This just shows an example of some oxygen and pH sensors on the device. And we can deposit these in different ways, but one we've used a lot is to use a piezoelectric dispenser to dispense these different materials onto the chip and into the different wells. We had this instrument made for us to do that. It's sort of called, we named it the Rainmaker microarrayer. And this shows some of the oxygen and pH sensors as they're arrayed on a device. CBDA is our lab name, Center for Biosignatures Discovery Automation. So where we are now is these extracellular sensors and these intracellular sensors, and what we're working on is being able to do as many of these in parallel as possible so that we can do the multi-parameter measurements, and that's also very challenging. And I'm just going to quickly go through some of these so you can just see what these response curves look like for these different sensors. This is ratiometric, so we get the response here in the oxygen sensor, pH and the correspondence to different pHs, dual oxygen and pH, so those we can use together, a glucose sensor, and then extracellular potassium, it's a green emitter. And what was important here, I have a really expert guy. Don't ask me all the details on this because I'm not the one that designs these, but I have a really good material scientist in my lab. And he's the creator of these different sensors. But this one, we had to create something. The commercial ones responded to sodium, and we couldn't have that. So he made this be much more highly selective. And the other thing is that we can't typically just use off-the-shelf, like molecular probes or something, because we're working at the single cell level, and the commercially available ones just don't work for what we're trying to do. So we're really having to create our own sensors. Then the intracellular one, this is a red emitter, and what's nice about this is it's cell permeable, and it distributes in the cytoplasm area. It goes in rather quickly. So that's been really good. And you can see here, we demonstrate where it goes in there. And we can also use pH intracellularly, and it goes into the lysosome, so, and it expresses at the same pH as the lysosome. So it's we call it a lysol sensor. So it's using the pH intracellularly as a lysol sensor. And that's just demonstrating that they overlap in the same areas. So what we created was this sensor, which is the SM2. We have an ATP sensor, which is highly selective, among other biophosphates. And this one internalizes rapidly in less than a minute. And this shows the blue and green ones, and then the overlay of that. So that's the lysol tracker there, and they all overlay in the same area. And then we've shown that we can use the APT sensor and done the measurements, the experiments, to show that we get the ATP sensor signal when we don't inhibit the ATP production, whereas here it was inhibited and we don't get the response. So we demonstrated that that's working as an ATP sensor. These show oxygen consumption on individual cells. The CPA and CPC, those are the Barrett's epithelial cells, metaplastic and dysplastic. And then these are breast cancer, normal, and tumorigenic. So we have this data. We're at the stage now where we're really trying to understand what it all means. And that's not easy to figure out biologically why is there all this heterogeneity and how do these things change. And I'll show you what we're going towards are these cell-cell interaction studies, because first of all, we can learn a lot from looking at an individual cell. In fact, identify mechanisms that don't appear when you're doing averaging of bulk cell populations, which there have been numerous examples showing that mechanism could be missed if you just look at the average. But it is important because most cells don't work in an isolated single cell environment. So we learn as much as we can from doing the single cell studies, but then go to cell-cell studies. So here we can mix different kinds, all CPD cells or a mixture of normal and CPD or whatever, and look at their oxygen consumption rates. And what we do see here is how it increases here with the decreases with the 3 cells versus when we have 1 or 2. So there's definitely something going on when you go from 1, 2, and 3 cells, and what is it? And here again is the isolated cells and then interacting 1, 2, and 3. So trying to understand what those differences in the rate mean. And then looking at as many as we can parameters at a time, multi-parameter measurements. So we've done the dual oxygen pH, and we're working on the potassium and ATP. And I'll show you an example of this one where we can combine extracellular potassium and oxygen because they don't respond to each other. We can separate those. And then this is one where we have the oxygen consumption rate externally, but then we can use this JC1 dye to look at the mitochondrial membrane potential at the same time and see that as the mitochondrial membrane potential increases as the oxygen concentration decreases. So that's combining an intracellular, intra and an extracellular sensor. So that's some examples of some of the things on the live cell measurements. And then we want to take snapshots in time of the DNA, RNA, and proteins. So we can harvest these cells for further analysis. And what we're doing is single cell transcription profiling. So here we're looking, it's a qRT-PCR approach, and we're looking at the expression levels here of 3 genes from 14 different kinds of cells, and just looking at those levels. And then here it's looking at the expression level of 10 genes from a single cell. And so we're working on expanding that actually to a whole genome approach. Here's looking at a single cell, the CPA cell, and looking at the gene expression changes in response to hypoxia. So what we did is the green are the control. That's before we put it into a hypoxic condition. And then we had the cells be in hypoxia for 30 minutes and looked at the response. And these are all genes shown to be in the hypoxia response pathway. But the only one that really showed up regulation was this one. So that's curious also. What does that mean? And then with the same approach, we can actually measure the mitochondrial DNA copy numbers. So we found 1,000 in CPA cells and about 1,500 in the CPC cells. So we're getting all kinds of different quantitative information that we're now working with the biologists to map and understand the biological significance of these things. And what's next? With the cell physiology measurements, we're now developing a next generation system because the current one in our lab is not very high throughput, but it's been a great platform for developing the different sensor capabilities. But now we want to be able to run thousands of cells at a time. So this will run tens of thousands of cells at a time. It's not built and available for your use yet, but we hope it will be in about the next 6 months. And this is really important. Is so that we can get enough data on all these different parameters on the live cell measurements so that we can understand these different signatures. We call the device the Cellarium. It's a cell in an isolated environment, and we can make all these measurements on it. And this is work that is funded by the NIH Common Fund Links program. So the Common Fund are the programs that the NIH director supports with his funding. And LINKS is Library of Integrated Network-Based Cellular Signatures. And I think this will be really important. It's a matrix, and I'll show you that in just a second, of responses to different perturbagens. And so we got funded to develop one of the technologies on this program. And so with that, we're developing this next generation system. And also, this is what the matrix looks like right now. Perturbation, cell type, and then the phenotypic assay. And Harvard and the Broad are the 2 first ones on this, generating data from this from their existing technologies and their large-scale efforts that they have. And then I got funded in the second round, which the first round wasn't even really something open to us, I don't think. But it's because they were the large-scale efforts. But what they've done now in the second round is fund 4 groups in the country to develop technologies that would be capable of adding new data to this. Ours will be the first to add single-cell data and also live cell data. So we're adding dimensions to this that don't exist. And in the program, we're also, I don't know if you want to say forcing, encouraging partnerships with the people in the consortium that they call the LINKS consortium. And we have some really exciting projects already underway with some of the people in there that'll really take advantage of this technology for some new biological applications. And then just to round out the biosignature diagram, I did genomics briefly, proteomics, cellomics, imaging. So there's different ways that you can get at anatomical imaging. Really, you're after figuring out where the tumor is and is it growing over time. And I know there's some experts here on PET and MRI and so on. We work with people that are doing a lot of work in the CT scans area at Mount Sinai. And let's say you're able to find a nodule. You want to be able to take sections through there and decide, is this cancer? Well, it's not so easy. And what you need to do is be able to figure out if it's changing over time. So I don't know if you could tell from, say, one point in time to another if it's changing. It's actually quite difficult unless you start implementing computer imaging techniques to really quantify what's happening, the location and how different things are changing. So this is looking at that. So this group at Mount Sinai teams with someone from Cornell that's really good at a lot of this computer imaging here. These are some of their videos showing also other features here on the tumors and tumor genesis. And then this is using their imaging techniques to determine if there's changes here between time 1 and time 2. And their algorithms and so on said, yes, in this particular case, the tumor is growing. So that's just another bit of information that's typically further on, but these earlier stage techniques like the cell CT will say, yes, this person has lung cancer, but it's not going to tell you where that lung cancer is. In fact, it's too early, at least now, to figure out where there may be some tumor in very early stages of growth, or maybe it's pre-tumor stage. So just to remind you again, this whole pathway here, there's a ton to do for the biosignature discovery part. And then, of course, all these other elements in the process, once you've figured out these different biosignatures, to actually standardize, qualify, and do the clinical validation is also a very huge undertaking. And you said we're after chronic health. And I think this is an approach that I think could be really promising. There's not only, as I said, the technology and implementation in that regard, but all the other aspects, healthcare, policy, insurance, and so on. But I've learned we do have discussions with insurance companies like Blue Cross Blue Shield. We've also talked with Kaiser. Blue Cross Blue Shield is actually investing in new technologies because they see the benefit for them in terms of having better ways to diagnose. Kaiser is a really great example. They're often early adopters of new technologies because the way their pay system is set up, it's to their benefit to have healthy members of Kaiser. In fact, and we know someone that's very high up, retired at Kaiser, that's going to be helping us get some of these trials done using the Kaiser system, which is also really good, of great value. I know I don't have much time if you want to ask me any questions. I just wanted to briefly show you where are we going with all of this in terms of some of the cell work. And about a week ago, I was at the NIH for a Common Fund workshop on single-cell analysis. So that's another program separate from LINKS, where they're investing because they see single-cell research being important for things across NIH and not something that would necessarily be picked up by one institute. Although we were lucky because our Center of Excellence in Genomic Science was funded by the National Human Genome Research Institute, and that was because of that unique program, the Center of Excellence in Genomic Science, where they were keen on taking risky new approaches to studying things related to genomics. But these were the questions they asked us at this workshop. The biggest scientific challenge in single-cell analysis work is significant advancements in single-cell work could be achieved with the invention of, and if the NIH gave me $1 million, well, most people said $1 million won't be enough, of course, but I would use it to do X, Y, and Z. So I won't really show you my complete responses, but just say a few things that I think relate to some of the things I already talked about. I already said how it's important to have this cell-cell interaction as part of it, but I'd say a lot of this single-cell research needs to head towards taking into consideration the whole tumor microenvironment. We can still, I believe, still learn a lot from this true single-cell isolated in vitro experiments, but we have to also be taking into account the tumor microenvironment. This shows how heterogeneous a tumor is. These are the blue are the capillaries. This is hypoxia in red, severe hypoxia in green, and then necrosis. That's a very hard thing to analyze. And then what conversations are being had in all these different pathways between cells and within cells. Cell-cell interactions really do matter. You saw just in 1, 2, and 3 cells how the data changed. So we're working right now on taking what we can do in single-cell physiology and integrating that with what we can learn from the biochemistry is one thing. And then also with the technology we're developing for the single-cell physiology that gives you the temporal information, we're developing the next generation cell CT for live cells. And so won't that be really cool to be able to correlate all those different kinds of measurements, the morphology measurements and the physiological measurements. And then ultimately to be able to do multi-parameter analysis of these cells functioning in their microenvironment. So this is where we're headed and a lot of the work that we're doing in our lab. And there's a lot of different people as part of the team. I mentioned a lot of our partners. And then also our funding has been predominantly from the NIH. The Keck Foundation is funding the next generation cell CT instrument for live cells. And then ASU is also supporting some of our efforts. And I didn't talk at all about our sensor bots or ocean work, but that's another area we could have another whole lecture on. So thank you.
Fascinating.
You're welcome.
Lecture. Open to questions. Rick?
On the sensors, you mentioned that they're highly selective. What's the mechanism that's giving that selectivity?
I don't think I can answer that. That it would be Yenqing in my lab. But it's the way he's designed the sensor and the reporter chemistry that he has so that it's very selective. And I don't know the specifics of it. I could get that information for you if you're interested or put you in touch with Yenqing. Yeah, I don't know. He does some really magical things with his materials developments. So. And actually, on our ATP sensor, we get that from the Gen group, and they won't even tell us what it is. So we're trying to figure that one out, because that has some really unique features, too. So.
I have 2 questions. Nature fools human quite often. Can 2 diseases have the same signatures?
I think
How will you distinguish between them?
I think that's a very good question, and I think there could be similarities or even same kinds of signatures. As we find out, also many diseases have many different forms. So, yes, I think we don't know enough.
May run into problems.
Well, I think the key thing is it's not so much what is that disease, honestly. It's what is it that's changing and going wrong in the system, and what can you do to put it on a normal course. And I think some of the changes we see could be representative of several different diseases. So the key thing is figuring out what's going wrong and how can we fix what's going wrong. We call things one disease, and it could be something entirely different.
The second question is the challenging task. How the changes are occurring within this cell. You can measure all that and put all the data on.
What's the fundamental mechanism for making the change? We are trying to understand that.
I see.
That is super.
Very important.
It's very important.
And that is the challenging intellectual challenge for students.
Well, so that's one reason to try to follow things over time, so you can see if you can figure out what is that mechanism that triggers something to go down one path versus another path.
That will take a long time.
I know. It's very challenging. And people kind of address it from different ways, and understandably, but if you could start, that's why I think it's important to have is multiple parameters and look at things over time and see if we can get a handle on where some of these mechanisms are.
Kinetics.
Other questions?
I have a quick question. So most cells exist basically in interaction with each other, and when you break them into a single cell level, is experimental artifacts a concern? Do they still represent their behavior in a system?
Yeah, we do tests on, is this a happy cell and is it normal according to what we know about those cells. We can do that especially in the cell lines. So there are certain things that you can measure. There are certainly things we're not measuring, and who knows, we might be perturbing something that we don't want to. But we try to keep the environment as controlled as possible and to keep the cells as happy as possible so that the measurements we're making are what we think we're making them on, and we keep them all pretty in a very controlled environment. But there's absolutely, that's why you have to go eventually from the single cell to the cell and eventually to the actual environment itself, because those are also important factors. But the goal is to learn as much as you can in these very controlled environments. If you stimulate this, what does it do to the cell? And to figure out those interactions.
Any last questions? If not, please join me in thanking Dr. Meldrum. This is a little memento.
Thank you.
It's
That's
set
very nice.
apart, so
Be
it's
careful.
apart.
Thank you.
But,
That's very pretty.
Commemorating your lecture today. Thank you.
Thank you. And thank you all for coming, and really a pleasure to be here. So thank you very much. Thanks.
And I think we have the box.
Oh. Oh, thank you.